I'm just going to call it, here's template, and here's the newly synthesized DNA pieces there, right? These are the Okazaki fragments, so this is the 3' end and the 5' end, and this is the 3' end and the 5' end, and this is the 3' end and the 5' end, and this part here is RNA, right? On each of these Okazaki fragments. So to get rid of them, what DNA polymerase has to do is get rid of that RNA, right? So what it's going to do is the repair enzyme, and I'll show you that in a minute, it's called DNA POL1, which is the repair enzyme. It's called POL1 because it was the first one discovered. But anyway, what it does is it sits on here, and it has a 5' and a 3' exonuclease activity. I'm going to exaggerate this, but it cuts out a nucleotide like this, right? So this RNA cuts off in this direction. Where's it going to add the new nucleotide? Right now there's a space in here, so there's a template down here. So it's going to pull a new nucleotide, right, into here, and it's going to put it in there. Where's it going to put it? On the same chain? What do you need for DNA synthesis? 3' hydroxyl, 5', 3', so it's going to add it onto here. Right? It's going to take one off of this Okazaki fragment and add the nucleotide onto here, one off of here and add it onto there. So you're getting rid of the RNA on this one, adding the extra DNA to this one. When it gets together, that's where you need your ligase. Yeah? Can you see that? Mm-hmm. It's pretty interesting how this stuff evolved. It turns out, this picture here, I think I'm going to show you later, but we'll give you the first idea here. Since this is from your textbook, I'll describe it to you. Right? The problem is, if you have a DNA polymerase complex here at this replication fork, it's moving this way to unwind the 2 DNA strands, right? How do you synthesize one this way and one that way? By the way, they're called bleeding strand. This one's called the bleeding strand, right? Because what happens is, as this fork's opening, it just adds nucleotide, right? Whereas this guy has to wait for 1,000 nucleotides before it goes. So the synthesis of this strand of DNA lags by about 1,000 nucleotides. Right? This guy goes first, and then this has to open it when that last one's made. So that's where leading and lagging strand get their name. How can this complex move through like that, this replication complex, and replicate both strands if one of them has to be backwards? Can it do it? Yes, it can, and this guy here. What you have to do, and I don't want to spend a ton of time on this, but if you take this strand, here's this lagging strand template, right? If you put a loop in it like this, what you've done is you take that lagging strand, and when you bend it around, it changes the direction, right? If this strand is 3' to 5, I'm going to do that chalk. If this part here is 5' to 3', and you're adding, right? If you take it and put a loop like this and still mark it, this is now 3' to 5' here, even though it's united, right? So you've taken a 5' to 3' and changed it to a 3' to 5' just by putting a loop in it. See that? So when the cell does that, when it loops it out like that, right, it makes the direction of synthesis the same way. So now the synthesis is going this way on this strand, right? This strand has been reversed, so it's now 3' to 5' in the template. The synthesis is 5' to 3' here, so there's that 3' hydroxyl. So as it opens up, it can synthesize both of those strands like that. You still have the lag, right, because this is separate. And you also have the problem that once this replicates like this, right, it has to, you notice that this is unwound here. This strand down here is unwound, that's the next template. It's going to build a primer there and synthesize that one next. But what happens is once this is done, this guy lets go, lets the loop flip around so that the next one is also in that same direction. What that means is the replication complex at one fork like that can all move in one direction down the strand as it opens, and there's not a lot of DNA sort of hanging loose, 1,000 nucleotides hanging loose while it's waiting to do this. Make sense? Question? Okay. So DNA polymerases, there's 3 of them in E. coli. They're called 1, 2, and 3. Polymerase 1 is a monomeric protein. It's a single protein. It has 3 activities in there, right? You can see here's the polymerizing activity. Where does it say that? So structural, the polymerizing rate. So it has a polymerization activity. There's the 3' to 5', that's the proofreading. And there's the 5' to 3' exonuclease, that's the repair. This guy has all of those activities. DNA polymerase 1 was the first polymerase discovered by Arthur Kornberg in 1957. And all he did is he isolated a protein from a ground up bunch of cells that could take nucleotides, ATP, magnesium, and a template, some DNA he threw in there, and synthesized DNA. And that was DNA polymerase. And he called it DNA polymerase, not DNA pol 1. And he thought it was the enzyme that really replicated all the DNA. But he did this experiment, right? He made an E. coli mutant. He thought he'd isolated a gene, and what you did in that day to prove that the protein or the gene or whatever did what you did is you made a mutant in E. coli that lacked that activity. So he took E. coli cells and he mutated the gene for DNA polymerase there and put them in a jar and tried to get them to grow, and the darn things grew. They grew just fine without DNA polymerase. Well, how can you replicate without DNA polymerase? Well, it turns out that the one that he found was the repair enzyme. The way you knew that is if you took these E. coli mutants that he had and you mutated the DNA with UV light. You shone UV light on them to put them, damage the DNA. Then you tried to get them to replicate. They wouldn't replicate. They couldn't grow at all because DNA polymerase 1 was needed for that repair, to repair UV damage. And it's also needed to do this activity there. There's two other polymerases. Pol 2, we don't know a whole lot about what it does. Pol 3 is the enzyme actually that does the bulk of the synthesis of DNA. So it replicates most of the genome. One way you can tell is if you look down here, right? This down here is called polymerization rate, right? And notice this guy's the one that does 1,000 nucleotides like I show you. It says 250 to 1,000. How can it be such a range? Why is it such a range? Is that real or are they just guessing? Is DNA polymerization 250 to 1,000 nucleotides? Actually, it is. Right? What makes the difference? Here's a great question. A great question. Doesn't have that many what? Nucleotides? No, close. It has to do with nucleotides. Yep. Sure. Remember the first step in all of this stuff that we have to do. It's good that you got that answer. First thing we have to do is unwind this DNA. When we unwind it, what are we doing? We're essentially pulling apart hydrogen bonds. We're melting hydrogen bonds. If you have AT base pairs, right, an adenosine and a thymidine base, how many hydrogen bonds? 2. If you have guanine and cytosine, how many hydrogen bonds? 3. So if you have a piece of DNA that's AT rich, then the polymerase complex can unwind it quickly and get 1,000 nucleotides per second. If it's GC rich, that's the hardest, right? It has all those triples to pull apart, and it only goes about 250. And in between depends on the content of whether you have AT or GC rich base pairs. So anyway, this one's much faster than the repair enzyme. Y'all see that? The other difference is this one has this property that we call processivity. 500,000, greater than 500 nucleotides. This enzyme here is only 3 to 200. What's processivity? Remember, most enzymatic activities, all the enzymes you talk with Michael Adams and everybody else, right? The enzyme picks up the substrates, which could be whatever. In this case, it's template and nucleotides, right? Biochemistry happens, the enzymology happens, and then they come apart. The product comes away. And then the enzymes have to find some new substrates again. Well, one of the substrates in this reaction is a template. And what DNA polymerase 3 has the ability to do is to get on that template and then stay on there, process down the template. So rather than binding, putting on a nucleotide, coming off, and then binding again, another molecule binding, one of them binds and it processes down there for a long time. And of course, this is just sort of like tunneling in a way, that when the enzyme becomes processive, it binds onto one of its substrate, which happens to be the template, and hangs onto it, then that makes the reaction go a lot quicker. So this guy is very processive because he catches on. This polymerase is much less processive, but it turns out it's plenty processive because the repair enzyme, the one that we described here, it only has to take out 10 nucleotides, right? And in most of the repairs where there's DNA damage, the eukaryotic guy, this guy, these are coli, so this guy only has to take out 12 nucleotides and fill it in, so 200 is plenty. That's plenty processed enough to do the repair, right? But not enough to make all the DNA. Okay, so this guy here, DNA polymerase 3, is very processive, it's very quick. The other thing is this has a single subunit. This guy has over 10 subunits. It's weird. How can you say it doesn't have 10 subunits that's over 10 subunits? It's hard because when you start purifying these guys, right, the difference of whether or not a subunit is part of the enzyme is a biological, a measure. You have to have an assay. You have to say, okay, this guy has 12 molecules associated with it. This molecule has 12 different proteins associated with it. If I take one away, then I lose my activity. So it becomes hard to determine exactly if you take one away and you lose 80% of your activity, or you lose 20% of your activity, is it a subunit or not? So it's somebody's guess. Okay, so let me go back here a little bit. We're going to jump back. This is the polymerase that's a single subunit, right? It has 3 activities, all 3 of those activities. And you notice that it has 2 different catalytic sites. It has one catalytic site, which is a site for polymerizing. So one of these is the template, right? This is the primer, this light pink guy's the primer, and then the dark pink's the thing. I guess that's what they're showing you. This is the strand that's being made because it's 5' 3-hydroxyl, adds that nucleotide there. Once the last nucleotide is there, it actually flips over to this other site, the 3' exonuclease site, has an activity site there, and it checks to see if it's the right nucleotide. This is not a great cartoon because it just shows a single strand of this. It doesn't check the nucleotide. It checks double stranded to see if it's hydrogen bonded or not. So it doesn't quite flip around. This whole thing has to move down. But the important thing is that what proofreading does is it increases the fidelity of DNA synthesis by about twofold, 2 orders of magnitude. Right? So DNA increases about 10 to the 2, so 100 times. It's 100 times less likely to make a mistake in DNA because it corrects its mistakes. And that's important. Okay. If you were going 1,000 nucleotides per second, right, and you didn't have a lot of fidelity, then every single wrong nucleotide you put in there would be a mistake. It would be a mutation in the DNA. And that could be disastrous to a cell. So this increases it that much anyway. This shows you, what am I showing you? So this is just showing you proofreading again. It looks to see if these guys are mismatched, there's a GT pair, there's no hydrogen bonds there, then what happens is this exonuclease 3' to 5' direction cuts it out and you lose that. So here's a diagram that shows you the subunit structure. Remember, there's about 10 subunits, I said. 2 DNA polymerase 3. Why is DNA polymerase 3? It's the one that does the mass amount of DNA synthesis. This enzyme wasn't discovered until several years after Kornberg discovered polymerase 1. And actually, the guy that discovered this started out being a musician. He was a cellist. He was pretty good because he got accepted to Juilliard. And he was at Juilliard studying cello, was in a tag football game, got tackled, and he broke all of his knuckles. And he couldn't play the cello anymore. So he decided, well, second best he'd do biochemistry, right? So he started looking for DNA polymerase, and he found another DNA polymerase because they knew that DNA polymerase 1 that Kornberg found couldn't synthesize DNA. What it was is a repair enzyme. So this guy was looking for the big one, and he eventually found it. And to me, the interesting thing about it is the guy who found this DNA polymerase 3 was Tom Kornberg, Arthur's son. So he discovered the real DNA polymerase. His father got the Nobel Prize for finding the one that did repair. And does anybody know of another Kornberg? There's Arthur, the father. There's Tom. He's the youngest son. The middle brother's Roger Kornberg. He didn't work on DNA synthesis. He works on transcription. He still does. He's at Stanford and he works on transcription. He got the Nobel Prize for doing transcription studies. He's one of the only people that ever get the Nobel, the father-son team, or the father-sibling team to get the Nobel Prize. Okay, so this guy has lots of subunits, and we'll talk about some of them along the way, probably not all of them. You notice here that it says that the alpha subunit has to do with polymerizing activity. That's the subunit that's involved in making that hydrogen, that phosphoester bond between the hydroxyl group and the phosphate group. One that I want to show you right now is this guy called the beta subunit. And the beta subunit is known as the sliding clamp. And it's the one that's involved in processivity. It's this clamp. It clamps onto the DNA so that this enzyme becomes processive. It can move up the DNA for at least 500,000 rounds of synthesis. Several of these, you notice, a lot of these guys down here they're part of the gamma complex, and what the gamma complex does is the gamma complex is what we call the loading complex, and its function is to put this clamp onto the DNA, to actually load it onto the DNA. And it does that in two places. first of all, this is the clamp, so